So have you all understood with the part fluid mosaic model? Anything you have not understood with that part? Hmm? Yes or no? Everything is understood? So let's see in the test part. But today's part also, I will take the test, right? First question. First question. I hope all the textbooks are closed. First question. The main component of cell membrane is. The main component of cell membrane is first cholesterol, second phospholipid, third carbohydrate, D it's sugar. A, okay, I'll repeat again. A cholesterol, B phospholipid, C cholesterol, carbohydrate, D sugar. Done. Second question. The prokaryotic cell does not have cholesterol. The prokaryotic cell does not have cholesterol, but have dash. I don't want any answers. The prokaryotic membrane does not have cholesterol, but has dash which will provide stability. But will have dash which will provide stability. A, stigmasterol, B, cephalin, C, hapnoids, D, both B and C. A, okay, the question is prokaryotic cells do not have cholesterol. The prokaryotic cells do not have cholesterol in their cell membrane, but they have dash which will maintain stability. A, stigmasterol, B, what was B? Cephalin, C, hapnoids, D, both B and C. Third question. 70% of protein. 70% of protein in cell wall is dash. 70% of protein, in cell membrane is dash. 70% of protein in cell membrane is dash. A, integral protein, B, intrinsic protein, C, extrinsic protein, D, both A and B. A, intrinsic protein, B, integral protein. Okay, A, integral protein, B, intrinsic protein, C, extrinsic protein, D, both A and B. Next, dash is also called as cell eating. Dash is also called as cell eating. Dash is also called as cell eating. Pinocytosis, phagocytosis, exocytosis, both B and C. Pinocytosis, phagocytosis, exocytosis, D, both B and C. Dash is also called as cell eating. Dash is also called as cell eating. A, pinocytosis, B, phagocytosis, C, exocytosis, D, both B and C. Why aren't you writing the test? Okay. Both are not there in biology. Done? Done? All the 5 questions are done? One is left, right? Oh. Next. Dash or extrinsic protein. Dash or extrinsic protein. I'll repeat. Dash or extrinsic protein. A, aquaporins. B, glycoporins. C, spectrins. D, antigens. Dash are extrinsic proteins. A, aquaporins. B, glycoporins. C, spectrin. D, antigens. Dash are extrinsic proteins. A, aquaporins. B, glycoporins. C, spectrin. D, antigens. Done? Everybody is done? Exchange among yourselves. Exchange. जिसको partner नहीं है आगे पीछे करो. But nobody will correct their own. Even if they write the answer, it's fine. Even if they write the option, it's fine. Is it clear?
Yeah.
Is it clear?
Yeah.
Okay. So first answer is phospholipids are majorly found. Option B. Option B. Second, which cholesterol? I mean, they don't have cholesterol. They have haploids. They have haploids. Prokaryotic cell have haploids. That option was C. That option was C. Right? Third question. What was it? Both A and B. Right? Why both A and B? Both have same name, intrinsic and sorry, A and B. Both A and B. D. The option is D. Integral and intrinsic is same.
So how is like A or B?
A and B I see. The option was D. Both A and B. Intrinsic is also called as integral proteins. It was C. First option was A, integral. B was intrinsic. C was extrinsic. D was both A and B. Right? 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 Am I right? A was integral. B was intrinsic. C was extrinsic. D was both A and B. So the answer is D. Is it clear? Fourth. Cell eating is also called as phagocytosis. Option was B. Last one, spectrin, right? C. Spectrin, C, extrinsic, right? Am I right? So I am passing this register. Please write your name. Please write your name. Write in questions you got correct on 4th, 5th and today. Do you remember how much you scored in 4th, 5th and 6th? Or should I pass the paper? Okay. Last one was spectrin. C. Am I clear? Please don't enter wrong marks, I mean wrong questions. Sorry, please don't mark the wrong. Please don't enter the wrong marks. I'll be checking off. If you don't remember, please ask. Okay. Write your name. You had answered your first test on 4th. Yesterday was 5th and today is 6th. Need repeater batch. Okay. So should we start with the part mitochondria? Mitochondria. Okay. See, 4th. Just write your how much you got correct questions. 5th, 6th. Any questions you have not understood for today's part? Anything you have not understood? So in short, if I say cell membrane is also called as plasma membrane, it is also called as plasmalemma, or it is also called as biomembrane. The most followed model is fluid mosaic model. The most followed model is fluid mosaic model, which says it is made up of 2 layers of lipid in which proteins are embedded. Right? The most found lipid is phospholipid. The most found lipid is phospholipid. What are the types of phospholipid found? Sphingolipids, phosphatidylcholine, phosphatidylserine. Okay, and that is also called as cephalin. Is it clear? Along with it, even cholesterol is found. What is the function of cholesterol? It will help in stability and making the cell rigid, making the cell membrane rigid. Is it clear? Is it clear? Cholesterol is not present in what is present? Sigmasterol. And in case of prokaryotes, it is haploids. In case of prokaryotes, it is. Am I clear? Am I clear? Anything not understood with this part? Then we had seen 2 different types of proteins, integral and extrinsic. Integral is also called as intrinsic. They are also called as transmembrane proteins because they form tunnels or they form transport channels through which material can pass. Remember, please remember, intrinsic proteins are also called as integral proteins, as well as they are called as transmembrane proteins. While extrinsic proteins, they are also called as external proteins, as well as they are also called as carriers. They are also called as carriers. Is it clear? Huh? Peripheral also. Is it clear? Should I move forward? Should we start with mitochondria? So, as you all know, what is mitochondria famous for? What do you mean by powerhouse of cell? It forms ATP as well as it stores ATP. Am I right? What is ATP? What do you mean by that? Why it is called energy molecule, or how does it give energy? Can you tell me what ATP will be made up of? Phosphate only? Glucose? Adenine attached to 3 phosphate groups. That is ATP. So when you break this bond, or you break this bond, or you break this bond, energy will be released. That energy we utilize for our daily work. Okay. Energy is stored. How energy is stored? Adenine, when it is bound to phosphate, it will require energy. When next phosphate is added, again it will require energy. When 3rd phosphate is added, again energy will be required. And this energy which is required will be this energy which is stored here, again it can be broken down and utilized. Am I clear? Am I clear? So mitochondria, it has lot amount of ATP which we can utilize. Okay. What else you know about mitochondria? Okay, powerhouse of the cell because it has ATP. Apart from it, it is a cell organelle. Apart from it. Oh, what do you mean by cellular respiration? Okay. So what is the purpose of you taking oxygen? Blood can be oxygenated. Why blood needs to be oxygenated? Why does organs require, I mean, see, in short, whatever oxygen you take, it goes to the cell. Why it goes to the cell? For respiration. What do you mean by respiration? Remember the oxygen which you are taking inside. The oxygen which you are taking inside eventually goes inside the cell, and the cell utilizes this oxygen to break down glucose. So breaking down of glucose and producing at last CO2 and oxygen along with ATP is called as cellular respiration. Am I clear? Taking in of oxygen inside the cell and utilizing that oxygen to break the glucose molecule and producing of ATP is called as cellular respiration. So in short, we require oxygen for breaking down of glucose. Why we need to break down glucose? To get ATP. Am I clear? Am I clear? Am I clear to all? My basic question is, how do plants respire? Or I'll tell the question. Do plants require oxygen? Yes. Plants require oxygen. For what? For what?
At night, there's no sunlight, so they can't undergo photosynthesis, so they have to respire.
Then daytime they don't respire.
No, they use sunlight and carbon dioxide to.
So you want to say that they take in carbon dioxide and give out oxygen, right? Nighttime then? My basic question is, like human, pay attention, last bench. आप first bench पे आके बैठा करो, please, please. आप प्लीज लास्ट बेंच पे बैठो ही मत, इधर आके फर्स्ट बेंच पे आ जाओ, इधर फर्स्ट बेंच खाली आ जाओ. Okay, my basic question is, like human, do plants also require oxygen to live on? Or do they also respire like yes, us? Remember one thing, let it be daytime or let it be nighttime, they also require oxygen. They also take in oxygen and give out carbon dioxide. But they do not leave it outside their body. They utilize that carbon dioxide for photosynthesis. We cannot utilize it for photosynthesis, therefore we throw out and plants utilize. So like human beings, even plants utilize oxygen to break down glucose and get energy. And when this carbon dioxide is released, they utilize it for photosynthesis. So it's not that they don't respire. They also respire by taking in oxygen and giving out carbon dioxide. But that carbon dioxide is utilized by them during daytime for photosynthesis. At night time, they release out that carbon dioxide as their photosynthesis is not performed. And therefore, at night time, you should not sleep below the plant because their carbon dioxide concentration goes high. And that may cause death of an individual because carbon dioxide concentration goes up and oxygen level goes down, which will cause carbon dioxide poisoning. Actually, it is carbon monoxide poisoning also, carbon dioxide poisoning, which will kill you. So there is no budni or anything like that below a tree. Especially they say don't sleep below, don't sleep below, under, not below, under. Which tree? Why? The reason is they produce a maximum amount of carbon dioxide at night. They produce maximum amount of carbon dioxide at night, which may lead to poison. Am I clear?
Wake up.
Huh?
It's like a big poison and can't wake up.
I did not understand. You are sleeping, right?
It's silent, it's suffocating.
So suffocating, you should go away from it. That's the thing. But see, we know that it causes poisoning. But there are laymen people who do not understand. They keep sleeping there. So that is the thing. There is no budni and all.
Actually, so photosynthesis doesn't replace respiration, just happens at the same time.
Yes. Remember, in case of plants, photosynthesis, respiration, continuously it is going on in the day. It does not mean that photosynthesis is on and respiration is off. Respiration is also on. Carbon dioxide which is produced, it will go for photosynthesis. At night time, at a night time, as photosynthesis, sunlight is not there, photosynthesis cannot take place, carbon dioxide concentration goes high. But they do take oxygen. They do take oxygen from the environment and survive throughout the night. Okay, so therefore there is no oxygen around the plant or very less oxygen around the plant at night. You should avoid going towards it. Okay. Is it clear? Is it clear? Okay. So in the case of mitochondria, the glucose is broken down. The glucose is broken down into smaller molecules and energy is released in the form of ATP. And ATP is present over there. Okay. So the main function of mitochondria is storage of ATP, cellular respiration and storage of ATP. Am I clear? Now, if you see the structure of mitochondria, it also has two membranes. Okay. Okay, so if you will see mitochondria, it has two membranes. One is outer membrane, inner is inner membrane. Now remember, outer membrane, it is smooth, but inner membrane, it has many infoldings. Infoldings means you can see inside, like these foldings are there. These foldings contain small particles like this, which are called as F1, F0 particles. And those infoldings which are there, they are called as cristae. They are called as cristae, Cristae. Am I clear? So mitochondria has two membranes, outer membrane and inner membrane. Inner membrane has many foldings which are called as cristae. Upon this cristae, there are F1, F0 particles, where mostly the cellular respiration will take place. Is it clear? Okay. The space inside is called as matrix. Well, okay. So there is matrix inside which contains mitochondrial DNA. There is a matrix inside which is called as, sorry, there's a space inside which is called as matrix. And inside that matrix, there is the DNA which is called as mitochondrial DNA. The DNA which is in the mitochondria, it is called as mitochondrial DNA. There are also ribosomes which are present. Am I clear? And those ribosomes are 70S type. Okay, they are 70S types of ribosome in the, okay, S is Swedberg unit. Okay, the unit you can say for ribosome, actually it's a protein, that is sedimentation coefficient, and for that the unit is Swedberg and it is written as S. Am I clear? Am I clear? Remember, mitochondria, it has two membranes, outer membrane and inner membrane. Outer membrane is smooth. Inner membrane has many infoldings, which are called as cristae. Which are called as? There are small particles on it, which are called as F10, okay, F1, F0 particles. Okay, the space inside is called as matrix. Okay, the between outer and inner membrane, the space is called as intermembrane space. But inside inner membrane, the space is called as matrix. The space is called as matrix has what? Mitochondrial DNA and 70S type of ribosomes. Am I clear? Am I clear to all? Is the structure clear to all? In F1, F0 particle or in crystal, cellular respiration will take place. Okay, in matrix as well as in crystal, cellular respiration will take place. Am I clear? Now remember, mitochondria. Mitochondria is known as semi-autonomous. It is known as semi-autonomous, self-replicating, self-replicating organelle. It is called as semi-autonomous, self-replicating organelle. Why semi-autonomous? Certain enzyme, it has its own DNA, so it can perform or it can form certain enzymes. But for some enzymes, it has to depend on nucleus. Therefore, it is called as semi-autonomous. Okay, it is called as semi-autonomous because some enzymes it can produce, but for some other, it has to depend on nucleus. Self-replicating means it will produce its copies, number of copies, depending on what the number of cell requires. So self-replicating means whenever the cell will require extra copy of mitochondria, it can produce on its own. It must not depend on anybody else. Okay, it is not dependent on anybody else or any other organelle. Is it clear why it is called a semi-autonomous self-replicating?
Cannot produce on its own, but can produce.
Right. Am I clear? Am I clear? Okay. So mitochondria, we'll just read the part. Everybody has entered the marks. So mitochondria. Kolliker first observed mitochondria as cytoplasmic granules in the striped muscles of insects. Atman established them as cell organelles and called them bioblasts. Why bioblast? Why do you think it must be called as bioblast? Was it blast? Then, because it produces energy. It produces energy for the cell, and therefore it was called as bioblast. Okay. Fleming and Altman were credited for the discovery of mitochondria. The term mitochondria was given by Carl Benn. Remember, discovery means that organelle was found out, and naming ceremony That is where, I mean, the naming ceremony was done by Carl Venda and it was discovered by Alsman and Fleming. Am I clear? Okay. F means first observed them in plants. Okay. It was first observed them, I mean, first it was observed in plants by okay. Korenberg and Mujbaum related them with cell respiration. I mean, the structure was found out, but what is its function? That function was discovered by Korenberg and Hofmann. Am I clear? Scavis call them as powerhouse of the cell. There are 1,000 to 16,000 per cell. Okay. Which cells will have maximum number of mitochondria? Huh? Okay. In case of animal or in case of humans, which cells will have, do you think? Okay, brain, liver, muscles. Why? They need lot of energy. So depending upon how much energy is required by the cell, that much number of mitochondria will be produced. Okay. So the cells performing maximum function, they require maximum energy and they will have maximum number of mitochondria. Am I clear? Heart will not produce as that because overall, if you see, heart is only involved in pumping, but the cardiac muscles which are there, that is performing the function. So therefore, it comes in the part of muscles. So muscles, liver, and brain, they have lot number of mitochondria. Am I clear? Only one in case of okay. Just in the case of protozoans, they have given the name as cholera and microstasis. They have only one mitochondria as they are very small and they are sedentary. Sedentary means they do not move from one point to another. Remember locomotion. When you're moving from one point to another, you require maximum amount of energy. As they live sedentary life, they are not moving from one point to another. They do not require much energy. So therefore, they do not require more number of mitochondria. 5 lakh mitochondria is in amoeba curioscius. Chondrium is the term given to the collection of all mitochondria in a cell. So collection of mitochondria is called as, usually plant cells have fewer mitochondria as compared to animal cell. Why? They do not, so as I told you, locomotion requires maximum amount of energy and they do not move, therefore they have fewer number of mitochondria. Am I clear? In higher animals, maximum mitochondria are found in flight muscles of birds. Why? Okay, flight, when they are moving from, or mostly you can say migratory birds, they cover around 100 kilometers per day sometimes. So remember, I mean, if they want to cover 100 kilometers per day, imagine how much energy they may be required. And continues.
What is the higher animal?
Higher animals. Now remember, there are two groups, invertebrates and vertebrates. So vertebrates are regarded as higher animals. Not in the case of height. Okay. Higher means which have more complex structures. So vertebrates are mostly regarded as higher animals. Not in the case of height. Please don't think that height is higher. Okay.
Vertebrates are lower.
Yes. Invertebrates are lower animals. So can you tell me invertebrates? Which are the invertebrates?
Spiders.
So insects.
And worms.
Huh?
Worms.
Now remember, even in case of certain invertebrates, they have complex structure. Okay. So that time even some, I mean in some textbooks, even they have considered them also as higher. But according to proper classification, if you say, higher animals are vertebrates. And invertebrates, as they don't have much complex structure, they are regarded as lower animals. Or you can say less complex animals. Is it clear? Mitochondria can make its shape as ellipsoidal oval, spherical, or depending, I mean, again, depending on what type is the shape of the cell, they will have different shapes. So structure, it has a covering of double unit membrane. Double unit means both the membranes are separated. Both are individuals. Okay, they are separated. Comparatively, the outer membrane contains more phospholipids. We have seen that in the case of cell membrane, again, there is phospholipids which is maximally found. Even over here, it's phospholipids which is maximally found. Am I clear? And cholesterol, then the inner membrane. Phospholipid present in the inner membrane is mainly diphosphatidyl-glycerol. Okay, diphosphatidyl-glycerol means it has two phosphate groups which are attached to glycerol. Okay, there are two phosphate groups which are attached to glycerol. And there are more proteins in the inner membrane. Remember, outside there are more amount of lipids. Outer membrane contain more amount of lipids. Okay, that is phospholipids. While the inner membrane contain more amount of proteins. I'll repeat again. Outer membrane contains more amount of lipids. Inner membrane. Am I clear? Outer membrane contains more amount of phospholipids or lipids, and inner membrane more amount of proteins. Each membrane is 60 to 75 Armstrong thick and separated by the space which is called as perimitochondrial space. Okay. Now remember, in some textbooks you will find perimitochondrial space, some textbooks you will find intermembrane space. You can see over here all. In the diagram it is intermembrane space. And in the wording form it is perimitochondrial space, but both mean the same. Okay. The space has many enzymes for the oxidation of fats and pyruvic acid. For the oxidation means for breaking down of fats and pyruvic acid. Remember, glucose is broken down into pyruvic acid, and pyruvic acid is finally broken down into CO2 and H2O. So in the matrix, in the mitochondrial matrix, glucose is broken down into pyruvic acid. And this pyruvic acid is further broken down in the in the intermembrane space. Then further breaking down is outside in the cytoplasm. Okay. The outer membrane of mitochondria is removed, and the structure is called as mitoplast. If there is no outer membrane, it is called as mitoplast. If there is no outer membrane, then it is called as, there is no outer membrane, then it is called as mitoplast. If there is outer membrane, it is called as mitoplast. That's the only difference. Outer membrane है तो mitochondria है. Inner membrane है तो it is called as mitoplast. Difference इतना ही है, outer membrane है या नहीं. Now, the outer surface, very important, the outer surface of inner membrane means this. The outer surface of inner membrane. Okay. The outer surface of inner membrane is called as C phase, while the inner membrane is called as M phase. This is called as which phase? This is called as C phase. This is called as M phase. Matrix के side वाला है M phase. Okay. Matrix के side वाला it is M phase. And जो outer membrane के side पे है, that is called as C phase. Is it clear? Membrane matrix की side वाला phase is called as M phase. And outer membrane की side वाला है, that is called as C phase. Is it clear? Some sessile particles attached to outer membrane are known as subunit of Parsons. Subunit of Parsons. Sessile means they cannot move from one point to another. Okay. What are their functions? Still not known, but they are present. Okay. Inner membrane has folding into number of finger-like projections, which I told you as cristae. I told you these projections which are there in foldings, they are called as cristae. And on this cristae, there is, you can see over there, ATPase molecule. Can you see over there? Structure of ATPase molecule. There is F1, F2 stop. F1, F2 stop. So that is actually F1. Actually, it is F below. You can see, oh, it's written as F1, F2 particle, but it's actually F0, F1 particle. Okay, they have not mentioned F0 over there. The term cristae was coined by Palade. Okay. In metabolically active mitochondria, the number of cristae is high. Means the cells which require more ATP, there the cristae number will be high. The inner membrane of mitochondria.
It says composed of ATPase. Does that case is unpacked? So does that like make ATP or does that break down into one?
I'll tell you that. Okay, let me come to that point. In the inner membrane, so sorry, where I was, okay. In the inner membrane of mitochondria, many electron carrier cytochromes are arranged in definite sequence, forming electron transport system for oxidative phosphorylation. Now, next when we'll see the respiration, that time I'll tell you the importance of it. But at present, just remember that the inner membrane contains this electron transport system, which is very important for respiration. Okay. Now, the inner membrane has studded with pin head particles called as oxysomes or elementary particles or F1, F0 particles. There in the textbook, it is given as F1, F0. Actually, it is F1, F0 particles. Okay, it is F1, F0 particles, which are also called as oxysomes or also called as elementary particles. Okay. Such types of particle was first found by Fernandez Morin. Okay. Now, very important, head of oxysome is composed of ATPase. Now, ATPase is the enzyme which is going to form ATP. ATP synthetase. Okay, synthetase means the enzyme which is going to form ATP or which is going to be involved in the last step in the formation of ATP. When I teach you respiration, you'll see that. Okay, so remember, very important, that F1, F0 particle contains the enzyme ATPase, or it is also called as ATP synthetase, which is important for formation of ATP. Am I clear? And is concerned with oxidative phosphorylation. Oxidative phosphorylation means, in short, breaking down of glucose and producing of ATP. The process in which cellular respiration is fine, but actual process after actual process in cellular respiration, it is oxidative phosphorylation, which means that ATP formation takes place with the help of oxidation process of glucose. Am I clear? So in which part of mitochondria does oxidative phosphorylation occur? In which part of mitochondria oxidative phosphorylation occurs? In a membrane, more specifically F1, F0 particles. Am I clear? Where is electron transport system present? Inner membrane. Where is electron transport system present? Inner membrane. F1, F0 particle is also called as well as oxysome. Am I clear? Am I clear? Okay. For the Krebs cycle, you all know what is Krebs cycle? What is Krebs cycle? Krebs cycle. Krebs cycle. Is it Krebs cycle, life cycle, or Krebs cycle? Okay. I don't, at present, I can just tell you that when glucose is broken down into CO2 and ATP, there are three steps. First is glycolysis. Second is Krebs cycle. Third is oxidative phosphorylation. So actually, when glucose is broken down, glycolysis, Krebs cycle, and oxidative phosphorylation. These are three processes involved. So the Krebs cycle occurs in the matrix. Oxidative phosphorylation occurs in the inner membrane. Am I clear? Am I clear? Oxidative phosphorylation occurs where? Where? And where does Krebs cycle occur? What is oxidative phosphorylation and Krebs cycle? When glucose is broken down. Okay, when glucose is broken down to CO2, there are three steps you can say. First is glycolysis, second is Krebs cycle, third is oxidative phosphorylation. So in short, Krebs cycle and oxidative phosphorylation are processes which are involved in breaking down of glucose. Am I clear? One to many double-stranded mainly one-to-many double-stranded circular DNA is present. Remember, in nucleus it is linear, but in mitochondria it is circular. Whereas circular DNA was found? Bacteria. In case of bacteria, mitochondria and chloroplast, circular DNA is found. While in nucleus it is linear. Am I clear? In nucleus it is linear. While mitochondria, chloroplast and bacteria, they have circular DNA. Am I clear? Am I clear? The DNA of mitochondria is GC rich. I mean, they have lot amount of guanine and cytosine. What is guanine and cytosine? Nitrogenous bases which are found in DNA. So the DNA is rich in guanine and cytosine. Mitochondria DNA code for the synthesis of 10 to 37 different types of proteins. Okay. The mitochondria DNA contains DNA polymerase and okay, we'll just at present if I'll say DNA polymerase. Okay, have you studied what is transcription and translation?
Yes.
What is transcription? When DNA codes for mRNA. When DNA codes for mRNA, it is called as transcription. And mRNA codes for protein, it is called as transcription. Okay. So when it is related, okay, what does DNA or do you know what is DNA replication?
Yes.
What is DNA replication? Replication of DNA. Okay. mRNA is messenger RNA. There are three types of RNA: mRNA, rRNA, and tRNA. So DNA, which codes for RNA, it can be mRNA, tRNA, and rRNA. That time it is transcription. Okay, you will be having this part very soon. Process of transcription, translation, and replication. Okay. So when replication occurs, it's DNA polymerases. And when transcription occurs, it is RNA polymerases which are involved. So mitochondria contains both DNA polymerase, okay, as well as RNA polymerase. Means what? They can perform its replication also of DNA, as well as it can perform its transcription also. As well as it has ribosomes, so it can perform translation. So therefore it is called as semi-autonomous. For certain things, it is dependent on nucleus. But as it has polymerase, DNA polymerase, RNA polymerase, and ribosomes, it can produce its own copies of DNA, its own copies of RNA, as well as certain proteins. Am I clear?
Yes.
Am I clear?
Yes.
What is the function of DNA polymerase? DNA polymerase will help in DNA polymerase will help in replication. RNA polymerase will help in transcription. Remember, DNA polymerase will help in replication, make copies of DNA. RNA polymerase will help in the formation of transcription. Formation of RNA. Transcription is formation of RNA. And from RNA, when proteins are formed, that time ribosomes. I'll repeat again. DNA replication, which enzyme? DNA replication, which enzyme? Transcription, protein, ribosomes. Am I clear? What is replication? Making copies of where DNA polymerase will function. Duplication, replication one. Okay, replication means making copies of DNA. Duplication means two copies of DNA. So it's one on here. But the actual, you don't say duplication, you say replication. Am I clear? So mitochondria of mammals have 55 S types of ribosomes. Okay, first it was predicted that there are 70 S type of ribosome, as I told you, 70 S type of ribosome, but mammals, that is animal cells, have 55 S. So except mammals, all have 70 S type of ribosomes. Is it clear? So what are the functions of mitochondria? Powerhouse of cell. Why? Because respiration occurs in mitochondria. Okay. Okay. Vitalogenesis, when you study reproduction, that time it is actually you add a vitelline membrane. One outside egg, there's a membrane which is added that is called as vitelline membrane. That process is called as vitalogenesis. Okay. So first thing, powerhouse of cell. Second, vitalogenesis. Okay, that is yolk formation and all of oocyte. And in cytoplasmic inheritance. What is cytoplasmic inheritance means? Have you studied Mendelian inheritance? Yes. What is pea plant was an example. Yes. Okay. So that is that is the Mendelian inheritance considers only chromosomes which are present in nucleus. Right? But cytoplasmic inheritance means it will consider even mitochondria. So when DNA is present in mitochondria or chloroplast, and it is specifying a particular protein, or you can say particular allele, that time it is called as cytoplasm. So suppose there are certain characteristics which are produced because of mitochondrial DNA. For example, if I say, just one example, if I say, the ears, okay, some men, they have hair on their ears, okay, it's because of these genes which are present on the mitochondrial DNA, not on nuclear DNA. They are present on mitochondrial DNA, okay, and they are expressed. That time it is called as cytoplasmic inheritance. Now, what is actually cytoplasmic inheritance is sperm and ovum both contribute nucleus, but cytoplasm, that is mitochondria, in case of plants it is chloroplast and all, it is contributed only and only by female. Male contributes only nucleus and therefore only nuclear DNA characteristics. When it comes to mitochondria, it's only female which is contributing. So all the mitochondrial genes or all the characteristics which are coming from mitochondria will be contributed by mother. So if the mother has genes for ears, hair, then of course all the males or all the sons who have developed from that mother will have that characteristic. So whatever mitochondrial genes a mother has, all that genes will be seen in all the children. Let it be male or female, because cytoplasm is contributed only by Mother. Remember, cytoplasm is only contributed by mother. So any mitochondrial disease is there. Any mitochondrial disease is there to the child. It is because of mother. Any mitochondrial disease is there to the child. It is because of mother and not father. So if the mother has any mitochondrial disease, she is advised not to give offspring or not to go for offspring, because compulsory it will be passed to their offspring.
It's always passed regardless of what happens.
Yes, it's always passed.
So between all the children, all of them will have the same. Okay.
Because mitochondria is contributed only by mother.
Okay.
Father will not contribute.
Genetic disorder.
Yes, it's a genetic disorder, but because of mitochondria. Am I clear? If the mother has any mitochondrial disease, she is advised not to go for offspring, because of course the mitochondria is only and only contributed by mother. So let it be any child who's going to. If it is nuclear, what will happen? One nucleus is contributed by mother, one is for father. If the father is having normal one, mother is having the carrier one, it can happen that father the dominant gene, mother's it is a recessive one. So which is going to be expressed? Recessive? No. Person will be not. But in case mitochondria, if the mother is suffering, both she's going to have recessive. From where dominant is going to come? Nowhere. So therefore,
If the father has a mitochondrial disease, it is not passed on.
no, because mitochondria is not contributing only. The sperm contributes only nucleus. The sperm contributes only nucleus and not mitochondria. Nothing, no Golgi apparatus, nothing. Save nuclear देता है. समझ में आया? Okay. Am I clear? So that is called a cytoplasmic inheritance. Am I clear?
Yes.
We'll stop here.
गांधी जी के गुण आ जाए Oh, I thought that's outside. केस के अंदर से बाहर नहीं
The chromatid, nucleotide, nuclear. So I'll revise with that part. First, with your doubt, if I start with. Yesterday we saw DNA. DNA, okay, is made up of what? Nucleotide. DNA is made up of here nucleotide. Which nucleotide? It is made up of what? Nitrogenous base. Nitrogenous base, that is adenine, guanine, cytosine, and thymine, right? Then phosphate group and deoxyribose. Is this clear? Is this clear till here? Now, these nucleotides, they form long chain. These nucleotides, so these are nucleotides, they form a long chain. Okay, it is called as polynucleotide. When this chain is just in a linear form, open form, completely open form, it is chromatid. This chromatid, it is not packed. It is just a thread-like structure. But when this chromatin, I told you yesterday, when it is packed around the stone, okay, this one unit, DNA plus stone, is called as. Okay, now understood? Nucleotide, nucleosome, and so chromatin is just a thread of DNA. But when this thread of DNA is wound around histone, that is nucleosome. And like this, chain of nucleosomes, okay, that will lead to the formation of Am I clear? Now, this, okay, one more very big difference I'll tell you. This nucleosome, actually, chromatin also contains histone around which DNA is formed. But this will be in the thread-like form. But when it is packed, it is going to be sorry. Okay, this will be just a thread like this. When it is packed completely into a small structure, that is microsome, like this one, linear fashion. It is called as when it is just like this, it's chromatin. when it is in the coiled form, it is chromosome. Am I clear? Tightly packed chromosome. Just thread-like structures, then it is chromosome. Understood? Nucleosome means histone found around. Is it clear? Anything else? Okay, should I move forward? Now, yesterday we had started with the part which is called as cell cycle. I told you yesterday that cell cycle means the complete life of a cell. It starts from growing of a cell, performing its function, and then but when we see related to cell cycle, we just calculate the or we just say that cell cycle has two parts. Interphase has interphase and cell division phase, which is also called as interphase, which is also called as preparatory phase, where it prepares itself for division. Preparatory phase, interphase means preparatory phase means it is preparing itself for division. Cell division is where actually cell divides to form either two or three. Depending cell division, there are two groups, meiosis and so you have complete process of mitosis, meiosis, you just have introduction. Okay. Okay. I don't understand the difference between homologous chromosome and sister chromatids. Joined by centromere, sister chromosomes. Three genes on the same locus, two chromosomes, one from mother, one from father, that is homologous. Is that part clear? Should I move that? So when you start with the interphase part, interphase, or you can say cell cycle. Cell cycle, maximum time spent in interphase. So suppose the cell has 24-hour life cycle. It spends around 21 hours, around 21 hours in interphase, and 3 hours in cell. Like it's your exam. You spend 21 days studying for the exam, and the exam gets over in 2 weeks. Why? Preparation needs more time as compared to examination. Cell division is examination time. Okay. You are also saying interface requires more time for preparation. Once it is completely sure that preparation is over, then only cell division will take place. So the cell spends maximum time in interphase. Okay. The cell spends maximum time in interphase. That is in the preparatory phase. Am I clear? So as I told you, mitosis is the equational division. While meiosis is reductional. Okay, here 2n number of chromosomes will produce 2n number of two cells. But over here, 2n number of chromosomes will be converted into n, and 4 cells will be formed. 4 cells or 4 daughter cells, here 2 daughters. So diploid cell will remain diploid only. But your diploid will get converted into Now, if you will see, in both the cases, that is, let it be meiosis or mitosis, interface is same. In both the cases, interface is same. What varies is actual cell division. We'll first start with interface and then we'll see. Don't understand, stop me at that point. Okay? Interface, again, it has three phases, or you can say three steps. First one is G1 phase, which is called as growth 1 phase. Second is S phase, which is called as synthesis phase. And G2 phase, which is also called as growth phase. Some cells have G0 phase. Okay, some cells have G0 phase, we'll see what it is. Now, in G1 phase, whatever things are required, things are required during S phase, that will happen in G1 phase. Before that, let me tell you, interface, two roles of interface. First thing, DNA replication. That is to make multiple copies of, to make duplication of DNA, or make extra copy of DNA. And second is organelle duplication. Why organelle should duplicate? Why organelle duplication is required? Because both the cells will get equal amount of organelles also. So overall, DNA replication and organelle duplication, or increasing the amount of DNA and RNA, that is the main work of interface. So that both these cells receive the equal amount of everything. Overall aim of interface is what? To increase the amount of DNA and also organ, so that both the cells receive equal amount of. Okay, now remember, in case of interface, S phase is very important. Synthesis phase is very important because your DNA replication is going to. That is, synthesis of DNA is going to take place. Okay, duplication, I can say yesterday I told you, when there is only one chromatic, two chromatic. So duplication of DNA is going to take place in S phase. So can you tell me what will be required for DNA replication? For DNA duplication, what will be required? Nucleotides. So in G1 phase, nucleotides will be formed. Whatever the enzymes are required, they will be. So preparatory phase for S phase is G1. जो भी कुछ S phase में लगने वाला है वो prepare करेंगे G1. So increase in the number of nucleotide. When the nucleotide is formed, enzymes required for nucleotide is formed, as well as duplication of mitochondria, chloroplast in case of plant cells, and other organelles will also. Okay, nucleotide formation, enzymes required for replication, mitochondria, chloroplast, amount will go on. Am I clear? In S phase, what is going to happen? Only and only DNA replication. That is synthesis of DNA replication. Last, in G2 phase, what will happen is only when DNA replication is over, G2 phase will start. In G2 phase, all the organelles will be duplicated. Here it is started, here it will get ended. So all the organelles will be duplicated and cell will prepare for cell division. whatever things like enzymes and everything, whatever will require for cell division, that will take place into this. Okay. Am I clear? Okay, so whatever will be required for cell division, it will form at the last 10 G. Now, remember some cells, they don't want to divide like neurons. Once they are formed, they will never ever divide. Those cells will go into the phase which is called as G0 phase. So once G1 phase is over, they will go to the G0 phase and they will never ever come back and resume to cell division. There are sometimes some cells are not getting proper nutrients or there is starvation period. That time also cells go to G0 phase. When they receive proper conditions, they come back to G0. So G0 phase is found in some cells like which once you enter into G0 phase, in case of neurons, you will not come back. But in some cells, if they resume back, they will resume in proper conditions. So during S phase, maximum time they will spend in S phase. Now remember, interphase is also called as resting phase. It is also called as resting phase, but actually it is not resting phase. Cell is not showing any growth, but cell is preparing itself for growth. That is, it is preparing itself for growth. So therefore, it is wrongly, it is not wrongly said, but it is not resting period actually, but it is a period where it is preparing itself for. Okay, it is also called as non-dividing phase. Well, it's not going to divide at that time. It's just going to prepare that. Am I clear? So I'll just go through your textbook, what it says quickly. What are the important points? So interphase, which is the phase during which cells will not divide, therefore it is called as non-dividing phase. Okay. So interphase, it is also called as resting phase. The interphase, it has three phases: first growth phase, synthesis phase, and second growth phase. In the first growth phase, RNA and proteins are synthesized and the volume of cytoplasm increases. Mitochondria and chloroplast divide. These two organisms have their own DNA. In late G1 phase, all cells must follow one of the two paths. They may either withdraw from the cell cycle and enter into R phase, okay, which is actually called as G0 phase. Okay, G0 phase is also called as R phase. Or they can go to the next step, that is synthesis. Okay. Synthesis phase, where chromosomes get duplicated. And second growth phase, where these cells, again RNA and proteins which are required for cell division will be prepared. And once G2 phase is over, it will be ready for cell division. This is what is given in the textbook. So formation of the new DNA. During the S phase of the interphase, each DNA molecule duplicates and repeats. So just remember DNA replication, that is formation of new. It's a very lengthy process. In short, I'm just explaining what your textbook says related to replication. In case of replication, we follow the model or we have semi-conservative type of replication. We have semi-conservative type of replication. Means there are two strands, okay? One is from 3 prime to 5 prime, another is 5 prime to 3 prime. Am I right? When replication takes place, they get separated from each other. They get separated from each other, and depending on what nucleotide is here, complementary to it, you will find the distance, which will be 5 prime to 3 prime. Over here also, depending on what complementary it is, they will synthesize. Suppose here it is A, here it will be. So here it is A, here it's going to be again, and here it will be, right? Because here it was, am I A or G or A and T? A and T. Right? Before it was here A and T. They got separated. A came here, T came. So A complementary T here, A complementary G. So two new, there will be two copies of DNA. Both they got separated and acted as a template to form. So here you can see two daughter cells, two daughter DNAs are formed. This will go in one cell, this will go in. So exact copies are going in both these cells. Why it is called semi-conservative? Because one is parent strand, one is core strand. One is parent strand, one is core strand. So therefore it is called as semi-conservative type of DNA. So in case of us or most of the living organisms, we have semi-conservative type of replication, where the daughter cells have one parent strand and one new. The daughter cells have one parent strand and one. Am I clear? Just write out. In case of many living organisms, semi-conservative type of replication, in case of many living organisms, semi-conservative type of replication, semi-conservative type of replication, semi-conservative type of replication is found. Semi-conservative type of replication is found where the two daughter cells form. The two daughter cells form. The two daughter cells form. The two daughter cells form contain each of one parent DNA strand, contain one of each parent DNA strand, and one old strand, and one old strand. One parent and one new strand. I'm so sorry. One parent and one new strand. Parent will be the old one, right? So in your textbook over here you can see this parent strand. One let's say 3 prime to 5 prime, another is 5 prime. Both the strands are getting separated, right? So the strand one is both will get separated and new one is forming. The one, new one is forming. Over here you can see this is the old strand, this is the new strand. Over here, this is the old strand. One will go in one side, this one will go in. Right here. Over here you can see this is the complete cell cycle. This is complete cell cycle where you will see maximum time the cell is spending in G1 is the longest one, S5 little small, G2. In G1 phase, what is happening? RNA and protein synthesized, which is required for Mitochondria divide, chloroplast. Whether it is all the organisms. In S phase, DNA is synthetic. That is chromosome get. In G2 phase, RNA and proteins which are required for cell division are and your smallest part is cell division. Smallest part is cell division. Am I clear? So now, can this cell cycle go on endlessly? Is it entire to several go on dividing? There is some point after which they will stop it. Now, in the case, for example, in case of plants, after many repeated divisions, the meristematic cells get converted to one. And once they are in permanent cells, they will not. Once they are permanent cells, they will not. Maybe in our case, it will be somatic cells. Somatic cells are normal cells. They will go on dividing many times. At one point, it will be such that it will stop dividing and it will be no longer. So in plants, the cells at the growing point meristem divide very rapidly and produce unique person. But once they attain a particular stage, and after this they cannot, they will get converted to germinal cells. Germinal cells are cells which give rise to ovaries. They give rise to egg and sperms. Okay, they only divide one and they will undergo meiosis and they will produce sperm and ova. They will fertilize into that cell. Now, there are some cells which are called as tumors. They have rapid cells. They divide so rapidly, and the processes which they perform, they go on whatever functions they have to perform. The processes they offers have. So because of that, they do not perform proper function and completely deform. So if you say you have tumor in head, there is tumor in head, brain tumor. That's why these cells divide so rapidly that there is mass of cells, but these cells do not perform. And therefore that person goes in. So even though there are many cells produced by, but they are not performing in job. And that may lead to production of cells, but no performing. Like yes, cancerous cells are what some of your normal cells are dividing so rapidly. There is no specific mediation. They divide so rapidly. Normally it should take 24 hours, but within in cancerous cells, within 3 hours this happens. Because they are dividing in 3 hours, does interface will take properly. If the interface requires completely 21 hours, but that thing is getting over within 2 hours. Then will replication take place properly? Will organisms form properly? So whatever the cells are formed, either they do not have proper organs, either they do not have proper DNA. So cells number is increased, but they are all the abnormal cells. And if there are abnormal cells, they perform the function. So only abnormal of cells, abnormality of the cells, but they cannot perform the function. And therefore the person, either it is brain tumor, the brain substance. When it is related to heart can happen, your cardiac muscle cancer, cardiac muscle is not working. Therefore, whatever structure is there, you can say cardiac muscles do not work properly, the complete body will. Even if you say example of any muscular tissue, okay. Suppose your hand muscular tissue, they have cancer, hand to know. So depending like this, various cancer cells are produced, and because of their attachment with other organs, they will be. So your cell production and okay. So as many cells are produced, as many cells are also. Because like if you say example, they have life cycle of 1 minute. They have life cycle of 1 minute. After 1 minute, they will die. So like this, the cycle will go on. production will be equivalent to time or cell into the when I clear this part, you are going to read or can I just this part of cell using that process. Before I can show the report, I just read on the report. So as I told you, cell division are of two types, mitosis and meiosis. Okay, both mitosis and meiosis, they have two phases. Karyokinesis, that is division of nucleus. Division of nucleus. Remember, karyon means nucleus. Kinesis means division. So, remember, mitosis as your syllabus has only mitosis, you just consider mitosis. So, mitosis first step is karyokinesis, where first nucleus will do. And second, it is cytokinesis, that is division of cytoplasm. So first nucleus will divide and then cytoplasm. Okay. So in case of mitosis, karyokinesis is followed by cytokinesis. In case of karyokinesis, again, there are four steps. First, prophase. Second, metaphase. Third, anaphase. And fourth, telophase. Okay, karyokinesis has two: prophase, metaphase, anaphase, and telophase. Okay, so in short, mitosis, first karyokinesis, that is division of nucleus. Cytokinesis, division of cytoplasm. So division of nucleus will occur in four steps: prophase, metaphase, anaphase, and telophase. Okay. So I'll just in short tell you what happens in nucleus. So in case of prophase, till now it was a thread-like structure chromatin. It will be condensed in prophase. Gets condensed with it will be easy to pass through the next. So therefore, in prophase, chromatin gets converted into chromosomes. First thing. Second thing, as it is division of nucleus, the nuclear membrane will, right? When the nuclear membrane will break off, more space will be available. So nuclear membrane will, there is a structure, small structure. Do you know what is nucleolus? Inside the nucleus, there is a structure called as nucleus, which is mostly involved in transcription. So nucleus is something. Okay. First thing, what will happen is nuclear membrane will break off. Nucleolus will disappear. Chromatin will get condensed to chromosomes. Okay. And one more thing, there is a structure which is called as centrosome, which is required for cell division. From that, spindle fibers will come. Spindle fibers are what? A thread-like structure which will pull the two chromosomes to the opposite. And when they pull the two chromosomes, here it will be separation of chromosomes. Then it will separate the two chromosomes. One will go there, one will. Oh, before that, remember, very important. In mitosis, the chromatids get separated. One chromosome has two sister chromatids. One will go at one pole, another will go to the other. So in mitosis, spindle fibers. This is one chromosome. Okay, centrosome will be present
two, one at this pole, one at another pole. From that, a thread-like structures come out, which are called as spindle fibers. From the centrosome, thread-like structures come out, which are called as spindle fibers. These spindle fibers bind to centromere at the point which is called as kinetochore. On centromere, there are proteins which are called as kinetochore, where the spindle fibers will bind and they will pull the histochromatids away from it. So this one will go here and this one will go. So in mitosis, the separation of sister chromatids. Separation of sister chromatids. So each one will receive one chromatid. Yesterday I showed this way. So as I told you, one, this is called as homologous chromosome, right? One you receive from mother, one you receive from father. This is parent cell. After replication, or you can say after interphase, each one will have sister chromatid. Each one will have sister chromatid, which will be exact property of this. Exact copy of this. Then what will happen? Spindle fiber, this will pull one, this will pull one. Over here also, this will pull one, that will pull one. And when they will get separated, each one will be that of that pair. One A, one B, one A. One A dash, one B dash. So they will be exact copies of their pair. Am I clear? So pulling of sister chromatid is done by spindle fibers. From where it is produced? Centrosomes. So in prophase, okay, right on. So in prophase, what is going to happen? Nuclear membrane will break. Nucleolus will disappear. Chromatin will get converted to chromosomes. And your centrosome will duplicate. Two copies to the opposite pole. Clear? Clear? Now, in metaphase, next phase, these all chromatids will get arranged at the center of this. Nuclear membrane has broken, therefore they can come out. So they will get all arranged at the center, which is called as M plate or cell plate in case of plants, or M line in case of animals. They will all get arranged at the center, and spindle fibers will start binding to them from the opposite side. One from this side, one from that side. Simple thing. Sister chromatids will get arranged at the center. Spindle fibers will start binding. At anaphase, they will get separate. Okay, they will get one will move this pole, one will go to another pole. And in telophase, they will complete this. Now remember, telophase will be exactly opposite of prophase. Where nuclear membrane will again start forming. Nucleolus will again come back. And chromosomes will then started forming prophase is exactly opposite of. Okay, so we'll see over here. So karyokinesis, there is now nucleus, prophase and prophase, metaphase and aphase. Remember P map. P and AP map. So prophase, the chromosomes are thick part, which will, the chromosomes become short and thick and they clearly divided by the. And you will see start forming chromosomes. Here they are spread like structures. Here you can see they are started forming chromosomes. Okay. So chromatin gets formed into chromosomes. Chromatids are attached at the centrosome. The centrosomes split into two along with simultaneous duplication of centrosome. And if there is a long structure centriole, and inside there is centriole. Okay, you can see over here. So there is one centriole. There is one centrosome which has been broken down into two, and they are moving to the opposite direction. Yeah. The centrosome split. Now remember, centrosome is only in animals. It is not there in plants. You see it only animals and not in plants. The centrosome splits into two along with duplication of centriole. Okay. Each centriole is surrounded by radiating rays which are called as asters. These asters are the ones which will be called as single fibers. The nuclear membrane and nucleus. The nucleated chromosome start moving towards the nuclear membrane. Now over here, this they are taken as I have taken as this side, they are taken as this. Okay. You are again this way and you are again this way. So over here, we are taken as the. And I here, and you will see here the two chromatids are together. In anaphase, what is happening? They are separated. They are moving towards opposite. In metaphase, their single fibers are just attached. But in anaphase, it is completely separated. And actually, that is early phase. Anaphase, they are completely separated. Telophase, again, the membrane is completed. Nucleolus is forming. They are again converted into thread like. And in cytokinesis, you will see cytoplasm will and some cells will be. So difference, remember, in case of animal cells, okay, in the case of animal cells, there is a furrow is given. But in case of plants, there is cell plate. Can you see the furrow? The plate is that is the difference. Remember, the animal cells, there is centrosome and your furrows. In case of plant cells, there is no centrosome and cytosome. This is the major difference in case of between and plant. Okay, so I repeat again. Remember, animal cell and plant cell in interface, chromatin is cell like. Once coming early process, the chromatin gets converted into chromatin gets converted into centrosome divides into two as well as centriole divided into two. Nuclear membrane starts this one. Nucleus also starts this one. Till the late prophase, what will happen? Centrosomes will go to the opposite. Centrosomes will go to the opposite pole and will start producing spindle fibers. Okay, but in phase, when people commanded by arranged hypercenter, spindle fibers will join to them. In the early anaphase, they will start separating, but by the late anaphase, they will get back again in telophase. What will happen? Nuclear membrane will form. Chromosomes will get converted into chromatin. Okay, and in cytokinesis, what will happen? Cell furrow will form in case of animal cell, and in case of plant cell, it will be cell. And therefore, these cells will be formed at the other. Am I clear? Have I understood? If you have not, we are just go through. So So I just finish this part. Significance of mitosis. Okay. Growth of the cell will divide, more number of cells will repair. If there is damage, birds, they are usually for repair. And replacement for the old one, when you are really. So asters are formed, asters are not. And asters in the case of animal cells, you will see asters will form the spindle fibers. But in plants, that is the spindle fiber. Okay. Your furrow is formed, your cytoplasm. It occurs in most tissues throughout the body. Your it only occurs in body. Remember, permanent cells are cells. They are not going to different. So in them, mitosis will not. And spindle fibers are formed with the help of asters, and there are no asters directly in spindle fibers are clear. So I think asexual reproduction. Asexual reproduction means over here, remember, organisms which are unicellular. They are only one cell. They will divide into the process by process of. Okay, and to maintain the same chromosome number is so if we go to this page number 70. If you have any doubt, when you want to give test of this chapter, try finish it tomorrow. Monday, if I will, it will be 25 marks test, 25 marks test, 1 hour. So that what will happen as we finish, we'll finish with this part. So you can just read if you have any doubts with clear tomorrow. I'll try to read the chapter tomorrow and test her.
अरे बताओ फिनिश पेज ये बे क्या किया तुमने ऊपर Mr. Classroom,